Hello, I'm Dr. Tim Sandle, and this is a presentation titled What's Lurking in the Water, and it's an exploration of pharmaceutical water system contamination. So this presentation is going to be looking at what we know about water system contamination. What are the characteristics of different water microorganisms? We're going to look at some published studies and assess what they tell us. We'll look at what influences recovery, because there are a number of variables that can affect that. We can also look at what we don't necessarily find, and this is a lead into viable but non-culturable microorganisms. And then we'll spend some time looking at some of the implications that studying the water microbiota signals to us, so things like biofilms, endotoxin, the necessity or otherwise for filtration, objectionable microorganisms like the Burkholderia cepacia complex, design factors, and aspects of the pharmaceutical dosage form like water activity. Okay, so what do we know? Well, the first thing is that our knowledge of water microorganisms, particularly in the pharmaceutical setting, is limited. And that's limited by the recovery being uneven, and the distribution of microorganisms in a water system is quite heavily skewed. It follows what we refer to as Poisson distribution. And it also stands that the bacterial counts don't necessarily fall into homogeneous groups. We're not achieving sort of a normal distributed populations. We're also going to get differences by divisions of the bacteria in terms of where they're coming from. So aquatic microorganisms are going to come from those that might originate from within the water system, particularly in the supplied mains water from the water company, the sort of natural organisms in the water system. And if they get transferred through into the pharmaceutical water system, then that is more likely to be a product of poor design. And then we've got organisms that could be introduced from the surrounding environment. So for example, when we turn off the water system, when it undergoes maintenance work, when we need to expand it by cutting into the system, then there's other types of organisms that can get in that are not necessarily part of the natural flora. So why should we spend time characterizing water microorganisms? Well, we can understand what's present in different water systems over time. We can see if there's a relationship between different water systems. So are we going to get contamination transfer from mains to purified and then maybe to water for injections? And this is all part of the kind of holistic approach to contamination control that's so important. We can build up vast libraries of data and trend over different periods of time to know what our normal variations are and to help us to detect when things are going wrong. And we can also understand what's more typical and what's atypical. And again, understanding the species of microorganisms can help us to understand when problems may be emerging. So it's also important to build up microbial libraries, so to take the most commonly occurring species and to put these into our culture collection and use those for things like testing culture media and for disinfectants. Now, in terms of what we might find when we start screening water systems, there are different studies. There's not too many studies really comprehensively looking at pharmaceuticals, but there are some. And looking at these, there's one from McAllister, for example, and one from Theresa. And these published reviews are signaling to us that pseudomonads and related organisms are the predominant species that we expect to find. Similarly, with a study from Kulikov as well. And here we get again the predominance of pseudomonads, Ralstonia, Flavobacterium. Bubbling under is Acinetobacter. And these are all what are characteristic gram-negative non-fermenting bacteria. And some of these organisms have distinctive patterns when it comes to resistance to different antimicrobial treatments. So some of them are organisms of concern. We also have a study by myself that was published about 10 years ago, and in this study I broke down the different contaminants into mains, purified water and water for injections. Now, mains water is that water provided by the utility company, a drinking water standard, and this water is supplied chlorinated, and then the pharmaceutical plant would proceed to remove the chlorine and soften that water through demineralization. The purified water was water produced by reverse osmosis, so it's using semi-permeable membranes with a substantial pressure, and the WFI was produced by distillation. And there was considerable diversity, but very similar in terms of the previous studies. So pseudomonads predominated. There were relatively high levels of Pseudomonas fluorescens, which can grow at ambient and lower temperatures. And this organism was found within the mains water, and probably was a facet of the water supplied by the utility company. It can run off from fields, for example. The second most predominant organism was Bravundomonas vesicularis, and this organism has an association with water and an ability to get associated with slime, and therefore there's a biofilm connection. And the third most common organism was Ralstonia pickettii, which is common to soil and water. It also stands that different types of water systems will recover different types of organisms, and there are variations that will influence what we find or don't find. So this can relate to the time that we did the sampling. So was it a time when production activity is at its height, or a time when things are relatively static, such as a shutdown? The volume that we sample is also important. We want at least 100 millilitres. The sampling technique to avoid cross-contamination by operators, and the sampling frequency, how often we sample and what that's based upon. Also, what we choose to characterise can be influenced by the heterotrophic plate counts that we are taking and how we are viewing those against predefined alert and action levels. But the types of agars used will bias towards heterotrophic bacteria, so that's those organisms that need an external carbon source. And pharmacopoeia dictates that we use R2A, reasoners 2A agar, and this is the agar medium that was especially formulated with low level nutrients to detect a higher proportion of stressed heterotrophic bacteria. So the theory runs that those organisms that are living in nutritively depleted conditions are more likely to grow on a culture medium that more closely matches those conditions. So comparative tests with higher nutrient agar show that R2A can yield higher counts over a 5 to 7 day period at a temperature of 20 to 28 degrees Celsius. There are other factors as well that are going to influence what we recover. So the subculture method is a factor. So there's more chance if we don't go from the low nutrient R2A straight onto a highly nutritive agar. So we don't go from R2A to TSA, but if we go from R2A to an intermediate medium like R3A, then that helps and provides a bridging step. Another factor is the identification method and what the database of that method actually contains. Another factor, and why something that influences the various studies that we've looked at, are geography. So geography is particularly influential on the supplied water to the pharmaceutical facility. So we can have nutrient poor conditions or oligotrophic conditions such as upland rivers, and here the microbial counts are within a few thousand per milliliter. Or we can have nutrient rich or eutrophic regions such as lowland rivers, and here the counts might be 1 million colony forming units per milliliter. It also stands that microorganisms in natural waters follow a seasonal distribution curve, and this is affected by the availability of nutrients and the temperature as well. And also the available carbon, or the assimilatable carbon, is also important, and this is generally at its maximum during the springtime. Other factors include water purification methods and so on. So what can we infer from this last review? So the inferences are that the water system will contain mainly gram-negative bacteria. There will be a relatively high level within the non-fermenter group in terms of taxonomic biodiversity, and we will expect to find very few gram-positive bacteria, and where we do, it's going to be those that are particularly hardy, like Bacillus species. It also stands that the size of the microorganisms that will recover will also vary according to the supply of nutrients, as well as other aspects of the microenvironment like pH, water flow, opportunities for attachment, and so on. It also stands that there's going to be some things that we just cannot detect, and that's because all culture media is selective. Most microbiological culture media is designed to select a wide population, but there's going to be limitations. And we're going to have limitations with detecting fastidious bacteria or those with specific physicochemical requirements. And it also stands that the water system will contain high levels of bacteria that are simply not culturable using standard methodologies. These are the so-termed viable but non-culturable bacteria. Some of these are going to be naturally non-culturable, and others are going to be non-culturable under a particular set of conditions due to the different environmental or nutritive stresses that they've been subjected to. It also stands that our water monitoring will have a bias towards those organisms that are in the planktonic or free-floating state, rather than those organisms that are in the benthic state, that is attached to a substratum or part of a biofilm community. And we know that there's variances by using alternative methods to assess microbial numbers in water, such as flow cytometry, and this will indicate that there are far greater numbers than the cultural methods suggest. And flow cytometry can detect a number of cells per sample, and alternatively we can also use other identification methods like 16S rRNA gene amplicon analysis, which also reveals a greater diversity in a water system than the standard laboratory culture methods can provide. Another thing is that simply recovering microorganisms from water doesn't necessarily mean that they will survive over the longer term. So survival tends to require a substrate for surface attachment, which is often a result of poor water system design, such as like reduced flow rate. We need to have suitable nutrients, although these can be at a very, very low level. There still needs to be some carbon source. The avoidance of excessive shear forces and other factors. Okay, so what can we make of all of this? Well, the first thing we need to be mindful of are biofilms. So many of the organisms that are indigenous to azeotrophic purified water environment are those that are adept at surviving to form biofilms. And Ralstonia pickettii, for example, is an associated community member of biofilms in pipes, along with non-fermenting gram-negative rods, particularly pseudomonads. And whilst the majority of bacteria are trapped in biofilm, biofilms are a problem because they're always constantly shedding clumps or individual cells, so we're going to get variable recovery patterns. So that's our first concern. Our second concern is with endotoxin. So high levels of gram-negative organisms are problematic in terms of endotoxin production. And it's been estimated that as few as 2.5 times 10 to the power of 2 cells of Burkholdia sapacea and 3.3 times 10 to the power of 3 CFU of Pseudomonas fluorescens is sufficient to give detectable endotoxin levels with the LAL test at levels of around 0.06 endotoxin units per milliliter. Now, endotoxin is either shed to a degree from viable cells during growth, or it's a function of non-viability when the cell has been damaged and broken. So endotoxin is a continued threat. Endotoxin comes from gram-negative bacteria. Gram-negative bacteria have a natural habitat in water. And the typical amounts of endotoxin associated with gram-negative bacteria is around 10 to the power of minus 15 grams per bacteria. So very, very tiny quantities when assessed by weight. And to give again another idea of how that's expressed, if you take the reference standard endotoxin that we use with the LAL test, then one endotoxin unit is based on around 2 times 10 to the minus 10 grams of lipopolysaccharide, which is roughly equivalent to a sufficient level of endotoxin. And if we take another measure, then if we have endotoxin in water, then that water will have been exposed to high levels of gram-negative bacteria at some point. So if we get endotoxin detection, there must have been a lot of gram-negative bacteria at some point in the past. Another thing about endotoxin is that the environmental endotoxin produced by gram-negative bacteria is highly heterogeneous. So the potency varies by bacterial species and strain, and there's also variations in solubility and by molecular weight. And the more potent endotoxins are those of the highest molecular lipid A weight and those that are most disaggregated. Now, endotoxin in water rarely represents the purified form of water that we'd use, say, in the LAL test. It's often combined with other materials and tends to form vesicles or membranous structures. And the size of these vesicles is dependent upon the type of lipopolysaccharide, the pH, the salt concentration, and the purity of the water. And this size then varies between 20,000 and 100,000 Daltons. And the larger these endotoxin aggregates, the more likely they are to be pulled towards surfaces. Another implication is that some of the more diminutive bacteria could potentially pose challenges to filtration within pharmaceuticals. So some of them, such as Ralstonia pickettii, could theoretically, under certain conditions, pass through very, very small pore filters. Now, whether this presents a major risk to sterile filtration, where a 0.22 micron membrane is used, is open to conjecture. but in some cases where there is sufficient concern, a 0.1 micron membrane might be appropriate one to use. Another factor that we need to grapple with is objectionable microorganisms, particularly when we're manufacturing non-sterile pharmaceuticals. And one of our concerns is with the Burkholderia sapacea complex, where there are related species and different bivalves of Burkholderia sapacea itself. And these are aerobic organisms found in soil and water, and they have a particularly renowned ability to survive in low nutrient environments for long periods of time that makes them a problem in pharmaceutical grade water systems. And the concern is that members of the BCC are human pathogens, and they can cause conditions like pneumonia in immunocompromised individuals, and therefore present a particular concern with inhalation products. So BCC organisms pose particular risk to water-based drugs, and the FDA in particular has warned drug manufacturers about Burkholderia sapacea complex, and there are various recalls on file that relate to that. So manufacturers need to establish procedures to address and control in relating to sanitary design, equipment cleaning, environmental monitoring, to prevent contamination, and also to undertake monitoring using appropriate agars. And there's the relatively new USP chapter that explains the testing regime. These water contamination risks also make us mindful about good design principles. To overcome the microbial challenges, then we need good system design, especially maintaining rapid turbulent flow and minimising opportunities for colonisation. And there's various factors listed on the slide, but these are subject of a separate presentation. We can also relate some of the organisms that we find to water activity, if we're using this as an expression in some non-sterile pharmaceutical products. So all pharmaceutical preparations contain water, and some of this water is free water, and other water is also there which isn't bound to the substance, and this available water is something that microorganisms can utilise. So water activity assesses how much free water there is, and it gives a measure of how much free water individual microorganisms might need. So we can have an understanding of what might be a risk in terms of potential product contamination, the level of water activity in our products, and whether we have a degree of confidence that those organisms would or would not survive. And we assess water activity as the partial vapour pressure of water in a substance over a standard state partial pressure of water itself. And the variables that can influence that include temperature, water purity, and humidity. So the higher the water activity of a substance, then the more likelihood is that it will support the growth of microorganisms. And there's some examples on the slide of different organisms, and you can see that the gram-negative risk, the Pseudomonad, needs much higher levels of water activity compared to the gram-positives, and then compared to certain fungi. But there may be variances within that we might have a particular gram-negative of concern, but we can use water activity to our advantage. With any assessment of water systems, it's important to undertake regular monitoring, both of the generation system, of user points, and particularly of supply and return. And we want to understand bioburden and endotoxin. And Annex 1 is directing us, for those involved in sterile products, towards regular assessments. And the inference there is that if the water is used every day, it should be monitored every day. And again, if we have 24-hour production, we need to make sure that we're doing different times of usage in order to get that full picture of the water system and any risks. And of course, it goes without saying that any results over action require investigation. And it's important not just to look at individual excursions from alert and action levels, but also to engage in trend monitoring. And trend charts are very useful in allowing us to deal with thousands of results, and they can help to show us variations in data, how variations change over time, which variations are expected and which are not, and what they might relate to things like maintenance work and water system sanitisation and breaking into the system and other things. How we can try then to divide between common causes and special causes. And examples of special causes can include a problem with a local outlet, problems following maintenance works, a new person starting sampling, a change to production usage. There's lots of variables that we can factor in. In terms of how to undertake trend analysis, well, something like MS Excel can be useful, but we can also use statistical packages as well. But it's useful to do things sometimes like convert the data, because if it's not following normal distribution, it doesn't always naturally fit trend charts, and we need to be careful when we're drawing out control levels and so on. So the data on the chart here, we've used the square root, and it's interesting because we can look at this chart and we can see upward movement and problematic trends, and hopefully we start react to these more quickly than we would if we weren't trending at all. Okay, so this brings the presentation to an end, and I've put some of the references on the slide. So these are some of the references to some of the papers that relate to the water microbiota characterization. If you want a copy of my paper, please contact me and I'll send you a copy. So in summary, what we've looked at, we've had a look at what we know and what we don't know. We've looked at some of the characteristics of water microorganisms. We've attempted to assess what these studies are telling us. We've looked at what influences what we find and what we don't find, and this is the whole viable but non-culturable and the seasonality factors. And then we've explored some of the implications that we can draw from this type of research. So we've considered things like biofilms, endotoxin, filtration, the Burkholder Cepacia complex and other objectionable organisms, design factors and water activity. So thank you very much for your attention. I'm Dr. Tim Sandle. If you've enjoyed this presentation and wish to see others and you haven't already subscribed to my YouTube channel, please do so. Thank you very much for your attention and goodbye.